Let's keep going then. You can think of questions and save them for the coffee break, for example, and come up and talk to the speakers, ask them. Our next speaker is Amir Shmuel from Montreal Neurologic Institute at McGill in Canada. So welcome, thank you.
Good morning, everybody. Are you uploading my slides? Yeah. I'm pressing. Thank you very much. So I'll be speaking this morning about the neurophysiological mechanisms underlying fMRI signals. fMRI is an indirect measure of neurophysiological activity. Now, most of us measure fMRI, but we are really interested in the changes in the neurophysiological activity. Therefore, the transformation between these two types of signal is of great importance. Here's an overview. I'll explain a little bit about the basics of intracranial neurophysiology, then on physiological mechanisms of bold response, negative bold response, resting state fluctuations in bold, and I'll end up by talking about the spatial specificity of the bold response relative to neurophysiological and metabolic activity. I have two disclaimers to say. First, I'll skip some slides that Rasmus Byrne talked about, and the other is that the field has grown. I'm going to show examples, mainly from my own research and laboratory, and I apologize to people that I do not present their results on the topic. So we'll start with basics of neurophysiology. Most of the studies with neurophysiology in live animals use extracellular recordings. You can see here the tip of an electrode, which doesn't penetrate the soma. It gets signals near the soma. The neuron is considered to be embedded in an extracellular medium that acts as a volume conductor. Now, when the membrane potential between two separate regions of a neuron is different, there is a flow, a primary flow of current within the neuron, which is being matched by a nullifying current in the opposite direction in the extracellular matrix. And that's what the electrode measures. So this is the mean extracellular potential. And usually in studies that, especially those that refer to neuronal correlates of functional MRI, this signal is being filtered into two main components. One is above 400 hertz, and this is the multi-unit activity, and the other is below 150 hertz, and that's the local field potential. Why is that justified? If we superimpose many spikes and we study the spectrum of action potentials, we see that most of the energy is around 1500 or 2000 hertz. In contrast, if we superimpose randomly on a time course simulated excitatory postsynaptic potentials, we see that the spectrum of the signal that we get has most of its energy in the low frequencies. Therefore, local field potential and multi-unit activity can be separated in the frequency domain, and local field potential usually is also separated to specific bands that are studied in the classical EEG literature. So components of the mean extracellular field potential, the multi-unit activity, a single event duration, the action potential is very brief, 1 millisecond. Local field potential, it's more like 10 to 100 milliseconds. That would be the EPSP. The multi-unit activity represents mainly action potentials of the projections neuron that form the output of a cortical area. And the local field potential represents the population synaptic, synchronized population synaptic potentials that form the input of a given area, including input from neighboring neurons. But this is the input, the postsynaptic potential. One thing which is extremely important to remember is that fMRI signal is an indicator of overall activity of very many neurons and processes. In a typical 3 Tesla voxel of 2 by 2 by 2 millimeters, there are 1 million of neurons and 10 to the 10th synapses. So we need to take that into account. We see the average of very many processes within each voxel. So just briefly about vascular mechanism of functional MRI. As mentioned by Rasmus, we measure content of deoxyhemoglobin. The signal is influenced, is sensitive to the content of deoxyhemoglobin, which is paramagnetic. Therefore, we get our signals from some kind of vessels. This is a scheme of cortical vasculature in the human brains from Duvernoy in 1981. So if we get functional MRI and we get signals, responses that are not diffuse over the entire brain, this means that there needs to be some control, some spatial control of where the blood flow, where the response goes to. This is thought to occur in the smooth muscles around arteries and arterioles, and also via pericytes that are cells that send processes around capillaries and near the junctions between arterioles and capillaries. And these processes can dilate or contract. BOLD is inversely proportional to the deoxyhemoglobin content. With increase in estrogen consumption, deoxyhemoglobin increases. With increases in arterial cerebral blood flow, deoxyhemoglobin decreases. And with increase in CBV, in cerebral blood volume, and nothing else, there is an increase in the content of deoxyhemoglobin, and therefore a reduction in the BOLD signal. Covered by Rasmus previously, the main chain of events in positive BOLD response. And now we get to the main part of the talk, relating metabolic, hemodynamic, and functional MRI signals to neuronal activity. As part of this presentation, I will use a system that I want to introduce now. That's optical imaging of blood oxygenation and cerebral blood volume. That's a system that we use mainly in animal models. It is composed of a CCD camera and illumination, usually with more than one wavelength of illumination. The physiological basis of this measurement system is identical to the physiology of functional MRI. The only difference is that functional MRI relies on the magnetic properties of deoxyhemoglobin compared to oxyhemoglobin, while optical imaging relies on the different absorption spectra of deoxy and oxyhemoglobin. So the physiology is just the same. We recently augmented, we upgraded the system to include also simultaneous measurements of membrane potential, of population membrane potential via voltage-sensitive dyes. So we can see here 2 cameras. Each of the cameras gets a mixture of the blood oxygenation signal and the voltage-sensitive dye signal, and via post-processing, modeling and post-processing, we can separate them. Now, I want to use this system to demonstrate temporal lag. BOLD lags behind neurophysiology, origin of BOLD, gray matter, vessels, which size of vessels, and the spatial specificity relative to the site of neuronal activity. So here it is. We already can see from the time courses below, this one of population membrane potential and this one of the hemodynamic blood oxygenation response. We can already see the differences in lag and the smoothness of the BOLD response relative to the impulse response, a very brief response of the membrane potential. Now, we look also at the videos, and please pay attention to which vessels carry the majority of deoxyhemoglobin, and also to the spatial specificity relative to the site of neurophysiology. So here, how do I start the video with this? Okay, so we'll have to skip it, but in any case, there is here a site of increase in neurophysiology measures because of FOPO stimulation in the rat somatosensory cortex, and then the majority of the signal can be seen in the pial blood vessel, also in gray matter, but really the higher amplitude in pial blood vessels, and the signal is carried out far away from the site that increased its neuronal activity. So we continue in relating neurophysiology to bold signal, and the next step is quantitative relationship, meaning can we describe a function? What is the function? Is it linear with each other? Bold is linear with increases in neurophysiology, maybe nonlinear, etc., etc. So here is a good old example of a study in which was performed in human subjects, functional MRI in human subjects, in the red dots, using a graded contrast of visual stimuli and measurements in V1, and then using the exact same stimuli to measure action potentials in monkey V1. So with increasing luminance contrast, we get a non-linear function of increase in fMRI and spike activity as a function of luminance contrast. However, the two functions are very similar, meaning that the positive bold response in human V1 are proportional to average firing rates in monkey V1. Here's an example of linearity and nonlinearity, this time a classic study from Lauritsen laboratory, who stimulated the climbing fibers of the cerebellum and used bipolar stimulation of the parallel fibers of the cerebellum. He used microelectrode to measure the activity of the Purkinje cells and laser Doppler probe to measure cerebellar blood flow. So when stimulating the climbing fibers, we see a linear relationship. When stimulating the parallel fibers, we see a sigmoidal relationship. So to conclude about quantitative relationship, the hemodynamic response is in many instances linear with the underlying neuronal spike activity. Non-linearities between hemodynamic response and local field potential or spiking activity have been observed. In the vast majority of cases, the hemodynamic and the bold response increase monotonously with increasing neurophysiological activity. So which type of neuronal activity is reflected by bold, spike or synaptic activity? That's a difficult question. The reason is that we already presented results showing that the positive bold is proportional to the activity of action potentials. However, action potentials are expected to be also proportional to the synaptic input, right? It's an input, synaptic activity, and output, the action potentials. So there are multiple variables here that are linear with each other. And correlation in this case does not actually allow us to conclude about causation. So in order to address this problem, we have to dissociate action potentials from cerebral blood flow or from bold signal. Dissociate spikes from local field potential or synaptic activity. And that's what Lauritsen did with the same setup that I presented a couple of slides before. When stimulating the parallel fibers, here's the stimulation, cerebral blood flow increased. However, the action potentials of the Purkinje cells diminished. This allowed Lauritsen to conclude that cerebellar blood flow depends on synaptic input to a region and not on the action potentials of the projections neuron in this region. In cortex, Logothetis and colleagues use simultaneous measurements of functional MRI and neurophysiology. We see here the electrode in the operculum in V1 of anesthetized monkeys. Here we see the functional MRI response to 12 seconds of stimulation, and we also see the fluctuations in the extracellular mean potentials that increase in amplitude upon stimulation. This is the envelope that represents the overall neurophysiological activity, and we see a sharp increase. Then we see the lagging behind, the smooth response, the bold response. Now, again, we need to somehow dissociate action potentials from synaptic activity in order to conclude what is the origin of the bold signal. So, to do that, Logothetis stimulated with 3 different periods. Epochs, 24 seconds, 12 seconds, and 4 seconds. And we can see that in pink, the bold response followed the duration of the visual stimulus, right? Long, more or less 12 seconds, and brief, bold response. In contrast, in blue and green, the action potential response was transient. It didn't follow the duration of the stimulus. In contrast to that, the local field potential response did follow the duration of the stimulus, and therefore it predicted the bold response better than the action potentials did. So we can conclude that the bold response reflects changes in local field potential or synaptic activity, which is input, but also local processing in a region more than multi-unit activity or output of a region. How about signal-to-noise ratio or contrast-to-noise ratio? Which of the signals has better contrast-to-noise ratio? From the same study of Logothetis, we see here a bold response, this time in this greenish cyan surface, to a stimulus that started at about 10 seconds here. So we see a sharp response upon presentation of the stimulus that reached about 60 standard deviations higher than the fluctuations of the physiological signal before the presentation of the stimulus. So obviously, the signal-to-noise ratio of the neurophysiology response is very high. In contrast, if we do the same exercise with the bold response, here in the blue curve, we see that after 6 seconds from the onset of stimulation, the bold reached approximately 2 standard deviations higher than the mean of its own baseline. So signal-to-noise ratio is very much in favor of neurophysiology. A signal-to-noise ratio of the neuronal signal is much higher than that of fMRI signals, and thresholding methods are likely to underestimate a great deal of actual neural activity related to a stimulus or a task. So, does bold always reflect changes in neuronal activity correctly? Well, perhaps not. Why? Because we also see negative bold responses. So what are these? Here's an example of my own study in human, where we can see a bold response, positive and negative, and cerebral blood flow response, positive and negative, in matching locations in response to partial visual field stimulation. So this can be interpreted as the origin of some vascular mechanism in which the increase in cerebral blood flow in the activated region with the stimulated region causes decreases in cerebral blood flow in adjacent regions, or it can be interpreted as reflecting with cerebral blood flow the decreases in neuronal activity which are induced via suppression from the active region, suppression of the non-active region, and therefore a decrease in cerebral blood flow. So in order to answer this question, I used the setup that was put together by Logothetis and colleagues. We see again the electrode here in V1 with the arrow, the green arrow represents the position of the electrode, and the green square represents the aggregate receptive fields of the neurons near the electrode. So we used two stimuli, one overlapped the receptive field and the other did not overlap with the receptive field. The overlapping stimulus induced positive bold response near the electrode. The non-overlapping stimulus induced negative bold response in the same location. We use the region of interest in order to sample the functional MRI response, and we can see here the signal, the positive bold response, and the negative bold response induced by the non-overlapping stimulus. Now, note that the negative bold response shows here a transient decrease and a gradual increase towards baseline, and then a post-stimulus overshoot. Similarly, the neurophysiological signal shows a transient sharp response, negative response, a gradual increase towards baseline, and a overshoot, post-stimulus overshoot. That's the mean power of the local field potential, and this would be the action potential response with similar time courses. So we can conclude that the negative bold response and the negative cerebral blood flow response in V1, in response to this stimulus, shows decreases in neuronal activity. And I'm being cautious here, and I'm saying in V1 using this stimulus because in order to generalize it over areas and tasks, more work needs to be done. Spontaneous fluctuations and functional connectivity fMRI signals are of increasing interest to the scientific community. So we know that we have large signal fluctuations in fMRI, and do they represent changes, fluctuations in neurophysiology? Now, this is a very important question because, as was shown earlier by Rasmus, there are also fluctuations, especially during the resting state, that can be attributed to systemic physiology. In this case, this is a beautiful functional connectivity or functional network that was obtained by Rasmus Birn simply by correlating the functional MRI signal with the respiration volume per time. Okay, so we get here a beautiful network. So we need to be cautious about that, and we need to check about neurophysiology. So again, the setup put together by Logothetis. Here is the electrode in V1, 7 sessions where the anesthetized paralyzed monkeys observed a green unchanging gray image, and 2 sessions in which they observed black and were in complete darkness. In order to be able to address the question of correlation between bold fluctuations in neurophysiology, we need to bring the sampling rate of the 2 systems, the 2 signals, to a common scale. So here is the functional MRI from 30 seconds, obtained at a TR of 1 second. That's the neurophysiology obtained at 21 kilohertz. We need to bring them together to a similar temporal scale. We do that by computing the spectrogram power as a function of frequency and time of the neurophysiology at a resolution of 1 second per sample. And this way, by integrating over the multi-unit activity domain and the gamma domain, we can get neurophysiology measures of population activity, overall neurophysiology measure at a sampling rate of 1 hertz, just like the sampling rate of functional MRI. Then we can pursue cross-correlation of the bold relative to neurophysiology with bold lagging behind the neurophysiology signal, and we get a function that looks like hemodynamic response function, statistically significant around the peak, which is obtained around 5 or 6 seconds, bold lagging behind neurophysiology. This can be seen in the broadband neurophysiology, gamma domain, multi-unit activity, and when counting action potentials, the action potential rate. Therefore, we can conclude that the 2 signals correlate. What about the spatial extent of the correlation? So here is the electrode in one position in V1, and we'll be able to see that the fMRI signal from a large region in V1, as far as the surface coil that we used was able to see, was sensitive to, was correlated to the slow fluctuations in neurophysiology. So here it is, 5 seconds and 6 seconds. And similar results were obtained later on by Shovnik et al. In the alert monkey that showed actually with the volume coil that the spatial extent of the correlation can be even larger and sometimes extend over half of the brain. So we can conclude that spontaneous fluctuations in fMRI signals in V1 correlate with locally measured fluctuations in local field potential gamma, multi-unit activity, and spike rate. The neural and hemodynamic signals are correlated over large regions of the visual cortex. Other cases in which a clear neuronal cause to changes in both signal cannot be found. So there are, here is an example from Sirotin and Das. What we see here are two signals. This is a response. So that's monkey visual cortex optical imaging followed subsequently by neurophysiology. So we can see here that the blood volume shows a very clear response to a visual stimulus, but in trials in which the primate was in darkness with no visual stimulus, just the pattern of trial after trial, the routine of the trial, created here changes in blood volume that were statistically significant. And so they concluded that local neuronal activity predicts visually driven stimulus, but not trial-related hemodynamics. Now, there were possible explanations that followed up these publications. They included neuronal changes simply missed by measurement or by analysis, attention that did not show clear changes in action potentials, the activity of neuromodulators that probably is the likely cause of this phenomenon, the activity of neuromodulators and especially the noradrenaline that is being sent to cortex when the monkey is alert during the trial and increases the noradrenaline in cortex and influences this way the vascular system. So under most of the conditions, but not always, fMRI signals are approximately proportional to the overall local neuronal activity. So I'll move on to the last topic here, spatial specificity of fMRI response relative to neuronal activity. So spatial specificity of the ball response is usually quantified by a point spread function, a Gaussian point spread function. Now, as indicated by several laboratories and ourselves too, it is not correct to just assume that one single Gaussian explains the variability and the spatial specificity in every single voxel. However, a point spread function is a good measure for the average over regions of the spatial specificity of functional MRI. So it is measured by full width half max of a Gaussian, and obviously we see here a narrow Gaussian and a wider Gaussian. So we will try to quantify that. In a previous study that I conducted, I used visual stimuli in the visual cortex, abutting visual stimuli, and based on the profile of the response at the end of the representation, the retinotopic representation of the two stimuli, we concluded that the point spread function in human V1 at 7 Tesla, we measured 2.34 millimeters and estimated that it is going to be less than 2 millimeters. Other studies before my study, published before my study, at 1.5 and 3 Tesla, estimated the point spread function at about 3.5 millimeter. Now, previous studies, including the study that I published on the point spread function, assumed a point-like neuronal response. However, neuronal responses are never point-like. Here's an example from Hubel and Wiesel, 1974, showing the scatter of receptive fields of one penetration in primate V1. So there is a scatter, which they estimated at the time at about 2 millimeter in a non-very quantitative manner, but obviously it is there. Can we verify these results? So here is an example from my laboratory using optical imaging and an electrode penetrating here. This is the region of interest around the electrode. And we used a series of 13 pairs of stimuli that abutted each other. Each of them actually was presented in a different time at a different presentation. But we had pairs of abutting stimuli. This way, we can compute very well where are the borders of the representation of these stimuli, and then we can measure the neurophysiology from the electrode in response to the stimuli, and also, obviously, the optical imaging. So here is the optical imaging response, and you can see that, as expected, the optical imaging response, the hemodynamic response, is not in a very sharp manner changing from response to no response, right? So there is here something like a sigmoidal function, but not the point that also gamma local field potential is not changing abruptly. There is a spread, and we want to quantify this change in hemodynamic response relative to gamma activity or relative to neurophysiology activity to get the pure spatial specificity of bold response, which is important for planning studies and modeling high-resolution imaging. So I'll present results on that at this point from humans. We previously published a model of a mathematical model of fMRI of cortical columns. This model addressed mainly, or was published mainly in the context of understanding decoding of fMRI signals from cortical columns with large voxels. We now fitted this model into ocular dominance data that I obtained together with Issa Yacoub at the University of Minnesota, both gradient echo and spin echo from human V1. We constrained the model by statistics, by measure, by parameters of ocular dominance from humans that we obtained from the literature based on post-mortem cytochrome oxidase histology from patients who lost one eye. And cytochrome oxidase is an enzyme of oxidative metabolism. If one eye is lost, then there is a difference in staining of cytochrome oxidase for one eye against the other. So we obtained information, we quantified information on real anatomy of ocular dominance into parameters of spatial frequency, irregularity, and branchiness, and included that in the model. Here is how it works, the fitting of the model to the data. We have here the data, the functional MRI of ocular dominance. We have here the model. The model produces many instances of anatomical functional organization of ocular dominance. It then convolves them with the hemodynamic point spread function, and it tries to match the result, the many results, to the data. What we get, or what we give, what we measure, is the probability of the data, probability of getting this data conditioned on the models and the parameters. So here is an example. At the end, we get a distribution of what we were interested in this study is to get the full width at half max of functional MRI. I'm afraid that the movie will not work here too. I'll give it a trial. No, it doesn't. But we have here the end result. The movie showed the convergence of the model, but here are the end results from two subjects. We see that the distribution of the point spread function values that can explain the data, given the model, here in spin echo, and here in gradient echo for the two subjects, and we get more or less results of, or approximately results of 1 millimeter in gradient echo and 0.8 in spin echo. So this is pretty good, and shows that given a well-designed pulse sequences and signal-to-noise ratio, and handling of the subject, we can get to a very good resolution with functional MRI. And with this optimistic mode, I would like to thank my colleagues, Denis Chaimov and Zeshan Yao from my lab, Issa Yakub and Camille Ogubil from the CMRR University of Minnesota. Thank you very much for your attention.